Today we are going to see the last topic, that is introduction to the genetics, which is an allied topic of the reproductive system. So in that we will see the chromosomes. Now, as we all know, genetic makeup of human being makes him or her different from others. So that all depends on the chromosomes. So all body cells, human body cells contains nucleus, which is an identical copy of the entire complement of the individual's genetic material, or that we can say a basis of the human body. Only exception in that is the RBCs, which does not have nucleus, and the gametes or sex cells. And in resting cell, the chromatin is diffuse and hard to see under the microscope. But when the cell prepares to divide, it is collected into highly visible, compact, sausage-shaped structures called chromosomes. Each chromosome is one of pair, one inherited from the mother and one from the father. So the human cell has 46 chromosomes or 23 pairs of chromosomes. Every cell has 23 pairs of chromosomes. A cell with 23 pairs of chromosomes is termed as a diploid cell because it will have one chromosome from mother, one from father. Now the gametes, that is the sperm and ova, with only half of the normal complement, that is they have 23 chromosomes each instead of the 46. So they are called as haploid, means the spermatozoa will have 23 chromosomes and the ovum will have 23 chromosomes, and therefore they are termed as haploids. And other cells which have 46 chromosomes, they are termed as diploid cells. Now the chromosomes which belong to the same pair have the same similar pair are called as homologous chromosomes, and the complete set of chromosomes from a cell is its karyotype. Here in this picture you can see the pairs of chromosomes. There are 23 pairs, 1, 2, 3, 4, 5 like this, up to 22, and the 23rd contains X and Y chromosome. So this is the sex chromosome. 23rd pair is the sex chromosome, which determines the sex of the person, that is male or female. And other 22 pairs are called as autosomes, and the 23rd is called as sex chromosome. Now this 23rd pair may not be the identical. Other 22 pairs are identical. They have the same chromosomes, but the 23rd pair may have different chromosomes, that is it may contain XX or XY. So the person having XX chromosome as in the 23rd pair will be a girl, and the person having XY pair in the 23rd pair will be a male or a boy. Here in this picture you can see the X and Y chromosomes. This is the X chromosome, female chromosome, which is bigger in size, and this is the Y chromosome or male chromosome. It is smaller in size. Now genes. So genes are present along the length of the chromosomes. All in the chromosome there is presence of the genes, and each gene contains information in code that allows the cell to make a specific protein. Means every cell in the body will have its specific function, and that function of the cell will be dependent on the protein which is prepared in that cell, and the preparation of that specific protein will be dependent on the genes in that cell. So the production of the cell will be a gene product, and each gene codes for one specific protein, and there are around 24,500 different proteins found in the humans. Now these genes normally exist in the pairs because each gene site is present at corresponding site on the both homologous chromosomes. So these genes will be in pair, and each gene will produce a type of protein. That protein will decide the function of that particular cell and therefore the particular tissue. Now comes DNA. DNA, we can say the very basic unit of the cell. Genes are composed of very long strands of DNA, and the total length of DNA in each cell is about a meter. One meter is the length of the DNA. So it is very long. DNA is a double-stranded molecule made up of two chains of nucleotides, and these nucleotides consist of three subunits, that is the sugar, phosphate group and a base. Here is the structure figure of the DNA. Strand 1 and strand 2. Two coiled strands are present, and there are the bases. We can see here guanine, cytosine, adenine and thymine. Now comes the genetic code. The function of DNA is to carry huge amount of information that determines all biological activities of an organism and which is transmitted from one generation to the next. So the key how this information is kept is found in the bases within DNA. These four bases are the adenine, guanine, thymine and cytosine. So the information is coded in the coded form in the DNA, and these four bases are responsible to preserve the information, and that is transmitted from generation to generation, and they are arranged in a sequence. Now the mutation means an alteration in the normal genetic makeup of a cell. So some mutations occur by chance because of the countless millions of DNA replications and cell divisions that occur normally throughout the life, whereas others may be caused by external factors such as X-rays, ultraviolet rays or exposure to certain chemicals. Example we can give of the cancer that is caused by mutation. Then comes protein synthesis. Now as we have earlier discussed that every cell it produces a specific protein which decides its function. So that protein synthesis is done in the within the cell and through a messenger RNA, because the DNA cannot move from its side, so it produces a RNA, ribose nucleic acid, or it is called as messenger RNA or mRNA. That is an intermediary molecule which carries the information for production of the proteins. Here we can see this is a DNA strand. mRNA is produced. The process of producing mRNA, it is a single stranded structure. That process is called as transcription. Means production of mRNA from DNA is called as transcription, and that mRNA then leaves this nucleus and comes into the cytoplasm of the cell, where there is protein synthesis. That mRNA gives the necessary information to the cytoplasm, and the amino acids which are present in the cytoplasm, they form a chain, specific chain which produces proteins. Specific proteins are produced. So thus mRNA is responsible for carrying the message from the DNA towards the cytoplasm of the cell to produce a particular form of proteins. So it is called as messenger RNA or mRNA. It is a single stranded, whereas DNA is double stranded. It contains ribose sugars and it uses uracil instead of thymine as a base. So one important feature of mRNA is it uses uracil as a base, whereas in DNA there is thymine. So So we have seen the process of transcription, production of mRNA from the DNA. mRNA is formed from the double-stranded DNA. Single-stranded mRNA is formed. Now the process of translation. Translation means synthesis of the final protein using the information carried on mRNA. It takes place on ribosomes in the cytoplasm and on a rough endoplasmic reticulum. So in this process the mRNA attaches to the ribosome, and the ribosome then reads the base sequence of the mRNA, and then there is production of the protein. Example, you can say that insulin is synthesized in pancreatic beta cells, is released into the bloodstream. So that is a production of insulin takes place through this process mRNA. Then cell division. We know that there is a continuous process of cell division, so our body grows. DNA replication. Now, DNA is the only biological molecule which is capable of the self-replication, and mistakes in copying may lead to production of non-functioning or poorly functional cells, or the cells which do not respond to the normal cell controls, and this could lead to the development of a tumor. Means if there is a mistake in the replication, then that will cause a tumor or non-functioning cells, or they may be over-functioning cells. Here we can see the DNA replication, original DNA, then there is addition of nucleotides, and again newly synthesized DNA strands are formed and they are coiled. Now in case of cell division, we know there are two processes, that is mitosis and meiosis. Mitosis is the process which produces two identical daughter cells from the one cell. Means both the cells which are produced from the daughter cell, they are identical in nature, and there is only production of two cells from one cell. That process is called as mitosis. Whereas meiosis produces gametes, and on fertilization, when the male gamete or sperm cell and the female gamete or ovum unite, the resulting zygote is diploid, because each gamete was haploid. That means the sperm cell will be haploid and the ovum will be haploid containing only 23 chromosomes. Unlike mitosis, meiosis involves two distinct cell division rather than one, whereas mitosis has direct division and meiosis has two steps. Meiosis produces 4 daughter cells, not 2, and all the 4 daughter cells are different from the parent cells and also from each other, and that is why that is the basis of genetic diversity and the uniqueness of each human individual. So as this meiosis takes place, the genetic characteristic of the person changes and it makes it unique. So here in this flow sheet we can see the mitosis and meiosis processes from parent cell. Here mitosis produces 2 diploid daughter cells. These are diploid, having 2 chromosomes, both derived from the parent cell. Whereas in meiosis there is exchange of the genes, because tightly fitting of genes, there may be exchange of genes. Now end of first meiotic division, there will be separation of these 2 genes, and then there is production of 4 haploid daughter cells from single daughter cell. So roughly we can say the mitosis produces 2 identical diploid daughter cells, whereas meiosis produces 4